Hi everyone, welcome to Reason with Science. I'm your host Jitendra. Our today's guest is Randy Schekman. He is an investigator of the Howard Hughes Medical Institute and a professor of cell and developmental biology in the Department of Molecular and Cell Biology at the University of California at Berkeley. He was awarded the Nobel Prize in Physiology or Medicine in 2013 for his role in revealing the machinery that regulates the transport and secretion of proteins in our cells. He shares the prize with James Rothman of Yale University and Thomas Südhof of Stanford University. In this conversation, we talk about his early career, membranes, exosomes, scientific publishing, and Elife. I hope you enjoyed the conversation. Thank you for listening. Hi, Randy. Welcome to today's conversation.
Pleased to be here, Jitendra.
Yeah, so today's generation, or the generation of 21st century, once they will grow up, of course, they'll be interested in learning why their lives first stopped in 2020 because of pandemic. But you were also interested as a child in microbes. So how did it start?
Well, I don't really know how it started. I grew up in Minnesota, but we moved to Southern California just around the time I was 10 or 11, just before that. And within a year, we moved, the family moved to our family home in Orange County, which at the time, early 1960s, was still almost rural, suburban, somewhat rural. And near my home, there was a dry riverbed with some sort of a sitting pond. And I remember one day collecting a jar of dirty pond scum from this river. And I had, from my birthday, I guess, a toy microscope. And I remember the fascination I had looking at these microorganisms that were swimming and crawling around on in a drop of this pond scum, things that you couldn't see with your naked eye. I just couldn't believe that that there was another world of equivalent complexity at this level. And somehow it captivated me, and I just continued to be fascinated by microorganisms really throughout my career. And that led to an interest in university. Initially, I was planning to go to medical school because I didn't know any better. But once I got to UCLA, I was able to work in a research laboratory in my first year, and that changed my life. The young professor with whom I was working, gave me a book that he said I really needed to read. It was called Molecular Biology of the Gene. It was the first book on the subject from James Watson, the very famous scientist, co-discoverer of the structure of DNA. And it was written in a style that was captivating. And I realized at that point that my life was going to be in basic science of cellular processes and not in medicine. And so for the rest of my time at UCLA, I worked in the lab and got deeply into studies on the mechanism of chromosome replication. And I was then fortunate to be able to study for my PhD with really the world leader in that subject, Arthur Kornberg, who won a Nobel Prize in 1959 for his discovery of an enzyme that copies DNA. And he was very instrumental in establishing my attitudes about how to approach a complicated problem using biochemistry. But I didn't like the competition in that field, so I chose an area that I felt would allow me to grow along with it, and that was biological membranes and how they get put together. And so for a postdoctoral period of time, I studied biological membranes. And then when I got to my current position at University of California, Berkeley, I started to explore the process of protein traffic and secretion, membrane assembly in baker's yeast, which was not really used as an organism to study secretion very much, but it turned out to be a good choice because yeast is an easy experimental organism given the application of classical genetics and biochemistry. And I was able to use both over the course of many years to uncover the mechanism of several steps in the process of protein secretion. So that's a thumbnail sketch of my career.
Yeah, it's impressive. Of course, we'll talk more about it in detail. I have a comment from from your early years when you started working on yeast. Yeasts have feelings too. So
that was a joke. That was a joke. Berkeley is a very liberal, politically liberal place. And I had a colleague some years ago who became an animal rights activist. And he invited these activists onto campus and they caused big disruption. So I, as a joke for one of my seminars, I took one of the pictures from the protest where it says animals have feelings too. And I changed the word from animal to yeast. Yeast have feelings too. But that was just a joke. But I showed that slide actually during my Nobel lecture, and some people thought it was a real picture. It wasn't.
Yeah, but that was funny. Yeah. But so once you started working on vesicular trafficking, what was already known in the field?
Well, the field had been formed through the pioneering work of early cell biologists, principally George Pilate, Keith Porter, and others, who developed techniques of microscopy to visualize organelles inside of eukaryotic cells, principally tissues in mammals, for instance, in the pancreas. And Pilate was really the pioneer, and he was able to show that there's a pathway of stations where proteins become transported across the membrane and then are encapsulated into a membrane vesicle and conveyed from point to point within the cell, eventually merging at the plasma membrane by fusion to secrete proteins to the cell exterior, proteins like insulin. And I got to know Dr. Pilate. Later, I never studied with him, but I grew very fond of him. He was a wonderful scholar, and deeply thoughtful person. And I remember hearing him give a lecture that was basically a reprise of his Nobel Prize lecture just after he won the prize in 1974. And I was just a beginning postdoc learning about membranes. And although the work that he did was pioneering, I was struck by the lack of any molecular understanding of this process. And so at that point, I resolved to study this mechanistically using yeast to isolate mutants, and then to study the genes and what they encode and how that informs the process of secretion. So he was absolutely instrumental in the field and in my career.
Yeah, and his work is so impressive. Once I looked at those images where those vesicles are budding, and he could show.
Yeah, his images are still among the best that have ever been published. He was a master craftsman of the electron microscope.
Yeah. So in general, in science, we work with a lot of metaphors. So how do you envision a cell when you are working with a cell?
Well, one metaphor that I've used is of an assembly line, like in an automobile assembly plant, where pieces are stamped out and things are added and moved along a conveyor belt, eventually to the finished product. It's a very loose metaphor for secretion, but it's similar. Proteins that are to be secreted are put onto the assembly line by being inserted into or through a membrane. This membrane is called the endoplasmic reticulum. And then these proteins become covalently modified. They become packaged into a vesicle, and this vesicle moves to another station where the contents are further modified and other vesicles are made. And so it's kind of like an assembly line process with certain rigid steps that can occur very reproducibly.
I mean, I think to understand the importance of this process, we can also zoom out a bit. So recently, Paul Nurse, another cell biologist, he published a book, What is Life? So in his book, he mentions one of the important properties of life as an information, where DNA serves as an information, and this information is translated into RNA or protein molecules. And so here, so if we look at in this picture, what do you think? Where does your work fits?
Well, certainly the work that we did relied on genes in the chromosomes that encode the building blocks and machinery that orchestrate the secretory pathway. But it's not all just in the genes. I have to say that I certainly agree with Paul, but the cell cannot form de novo. Even if you had, if you were able to, if you could read the entire sequence of a genome in any organism, it wouldn't tell you how that organizes the cell. The cell had to evolve from some primitive life form and then slowly build an apparatus that can be replicated, but which doesn't occur de novo at each generation. So and this was something that Palade recognized, that the endoplasmic reticulum is a template that is self-perpetuating. It does not form de novo. In fact, very few membranes form de novo. They have to form from some pre-existing organelle. And that is, obviously, it is the product of the genome, but it isn't reproduced by the genome every time a cell divides.
Exactly.
So there is a inherent structure to a cell that can actually continue to operate even after you remove the nucleus. There are experiments where you can take a cell and dissect out the nucleus, and the cell will continue to move, and it can for a while continue to manufacture new proteins and secrete proteins. It can continue. The secretory pathway operates even after you block protein synthesis. Membranes are still formed, and they are consumed, and they are recycled. That can continue very, very, very long period of time, even in the absence of protein synthesis. So this relies on having built a self-replicating machine that obviously is informed by the genome.
Yes. So if we think of origin of life, in even if we can go at the unicellular organism, and we think that in this early times there were like replicators in the soup.
Mm-hmm.
So what you are suggesting, that the membrane or in general, this encapsulation of this replicator can be another independent event.
Yeah.
Right.
We can only speculate, really. We haven't actually reproduced de novo the formation of a cell. Maybe in time that will be possible. But right now, that subject, although it's an active area of investigation, is all entirely speculative. It's unlike most experimental science, where you can design something to test a principle of life or a principle of the universe. With the origin of life, it's all entirely speculation. And maybe with time, someone will actually achieve a protocell that can self-replicate, but we're a long way off from that. So it's still be a matter of debate about how life originated. There are, of course, many people who think that life on Earth came from a primitive life form that landed on a meteorite from some other universe. But that just moves the problem back one step. It had to originate somewhere. And we don't really know where or how that happened.
Yes, but this sort of makes the membranes really interesting, right? That how they are formed and
well, membranes are, an ideal agent to encapsulate a cell, because they can form spontaneously. If you just take a solution of phospholipids in an organic solvent and you dry it down at the bottom of a tube, and then you resuspend it in buffer, and you shake the tube, vesicles form spontaneously because of the thermodynamic properties of the acyl chains on phospholipids. So that, that may have happened originally, that some replicating center was encapsulated within a membrane, some primitive membrane, and just enough may have been contained within this primitive vessel to allow the creation of a self-replicating cell. And the beauty will be if we can actually recapitulate that process in the laboratory. That has not happened yet.
Yes, but since you've been working with the different cellular organs which do have these membranes, so do they have like similarities, or they are different?
Well, the membranes that participate in the conveyance of secretory proteins share some properties, but they also are different. They have different protein composition, to some extent a different lipid composition. They are distinct, and they can be separated from one another by physical techniques, but they interrelate by a flow of material through the pathway, and so they have to have some way of achieving their unique composition, which is believed to happen by selective retrieval of certain proteins, such as those that are characteristic of the ER that happen to escape. They have to be retrieved. Likewise, within the Golgi, proteins that move through the Golgi occasionally carry along some Golgi resident proteins, and those have to be recycled. So there's an active forward and backward flow that establishes the equilibrium position and structure of each organelle.
Exactly, but the basic properties are preserved when we start from yeast and go to more complicated.
Yeah. Well, nature solved the problem of how to secrete soluble proteins 2 billion years ago, and the mechanism that evolved, it made sense to keep using the same parts, and so that of course evolution has changed many of them, and new decorations have been added, but the basic machinery has been preserved, and that's something that we learned when we established the pathway of secretion in yeast, that it was formerly the same as in human cells, and when we were able to identify genes and their counterparts were subsequently found in the human genome, it became perfectly obvious, not surprising, but perfectly obvious, that the genes are evolutionarily conserved. Paul Nurse had demonstrated that himself in his early work, where he was able to use a yeast mutant defective in cell division, and to repair the defect by fishing for the human gene equivalent. And he showed quite dramatically that the human gene for this protein could replace the yeast gene, and the yeast cell would survive as though it were using its own protein. So that made a very dramatic statement that genes of fundamental importance in intracellular events are functionally conserved. Many of them, not all of them, but many of them are functionally conserved, which is really it's really quite surprising, but there it is, it works.
Yes. So then let's talk about your first mutant, like the mutant that you guys discovered, SEC1. And there was this beautiful picture where the budding yeast, you could see those vesicles at the daughter's cell.
Yeah. Well, when we started my lab in 1976, I was not trained as a geneticist, so I didn't think that way, at least not initially. And when one of my first and most outstanding graduate students, Peter Novick, joined the lab, we looked for chemical inhibitors that would block secretion, with the hope that we could find something that would allow us to accumulate secretory vesicles and isolate them and study their composition. But the chemicals that were available to block secretion were not effective in yeast. And so we resorted to looking for mutants, even though that wasn't our first plan, but that worked out very well. And in late 1978, Peter obtained his first mutant. We assumed that these genes would be essential for cell growth because we imagined that the vesicles that carry secreted proteins into the bud for fusion and discharge to the cell exterior would also, of course, have membrane proteins that would become part of the plasma membrane, and thus the fusion of a vesicle at the cell surface would allow the surface of the daughter yeast cell to grow. And without that, of course, the cell would not grow and would die. So we looked for what are called temperature sensitive conditional mutants, that is, mutations that change an amino acid in the folded structure of protein that render the protein thermally unstable. And this is a common phenotype for point mutations. If they change the stable folded structure of protein, they become somewhat more thermally unstable. And so this is a classic way of defining essential genes in a microorganism. And we did so, and we found the first mutant, SEC1. And in the spring of the spring of 1978, George Palade, whom I've talked about, came to Berkeley to give some honorific lectures. And although I knew him, he wasn't aware of my new work on yeast, so I was excited to tell him about that. But the students organized a dinner, and Peter Novick told him about this mutant SEC1, but this was before we had looked at the mutant by electron microscopy. And so Palade, of course, encouraged Peter to do that. And so he went ahead and did that. And by the summer of 1978, we had the images that you've described, where the cells under normal conditions have just a small collection of vesicles that are underneath the bud portion of the growing cell. But when you block that last step with this mutant SEC1, vesicles fill up the entire cytoplasmic compartment, and eventually the cell chokes because it can no longer grow and it's still making all of this stuff. So that was a revelatory moment, really sort of the eureka moment in my career to see that image. And I knew from that moment that we would have wonderful experiences over the next 20 years figuring it all out. So Peter then, of course, launched into isolating many more such mutants. And within 18 months, we were able to publish a paper where we described 23 new genes that inform this pathway. And all of the work that we've done since then was launched by that study.
Yeah, and then the same study basically led to the discovery of COP2 complex, which could show how these vesicles are formed.
Yeah.
Yeah.
Well, let me just digress on that point a moment. So I was trained as a biochemist, and the genetics for me was just a tool to discover the proteins. And a lot of yeast people stick with the genetics, and that makes it more difficult, I think, to understand molecular mechanisms, because when you're studying a new pathway, such as we were, I felt that if we clone the genes, I mean, one could do that at that point, that the sequence of the genes wouldn't necessarily be that instructive. These would be mostly new genes, and they'd just be a string of amino acids, which wouldn't tell you what the protein did. Now, of course, there were some exceptions to that, but for the most part, the genes that are required for vesicle budding showed no homology to anything until the human genome was sequenced subsequently. And then you could see that there were the same genes, but that still didn't tell you what the proteins did. So I felt all along that one would have to have some biochemical approach using the mutants to help us focus on the physiologically relevant pathway. So we established, subsequent to Novick's work, I had another wonderful graduate student, David Baker, who's gone on to his own very successful career in protein design. he devised a cell-free reaction, a way of breaking open yeast cells that reproduced in the test tube, essentially the first half of the secretory pathway. And we use that to, as an assay to isolate functional forms, which then led to the purification of the proteins that constitute a coat that envelops and pinches a vesicle from the endoplasmic reticulum membrane.
Yeah. But what is so special about this COP2 complex?
Well, it has many interesting features. One is it's easily assembled and disassembled. And when it assembles, it has the intrinsic ability, because of several of the subunits, to recognize cargo proteins, membrane proteins that are on the cytoplasmic surface of the ER that are destined to be captured and moved to the Golgi apparatus. And these are distinguished from the many proteins that are designed to stay and function in the ER. They are left behind. And so the COP2 coat not only grabs the right cargo, but it then pinches that cargo into a vesicle that buds from the ER. And then the coat spontaneously dissolves, leaving a strip vesicle that is then targeted for fusion to the Golgi apparatus, and the subunits of the COP2 coat can be recycled to perform another budding event.
Yeah. And then finally, these vesicles, once they are released from the cell, they can act as both long-range communicators or short-range communicators,
Sure.
which makes it very important in terms of animal physiology, right?
Sure. No, these secretion is an essential aspect of physiology, and it informs processes in essentially all of our cells, and it's most active in the brain, organizing the secretion of chemical neurotransmitters. The same SEC1 protein that we discovered in yeast that's required for the fusion of a vesicle at the in the yeast bud, in the daughter cell bud, is involved in the fusion of synaptic vesicles to the presynaptic membrane of a nerve terminal. So, and that process occurs in many different stations in the cell and in essentially all cells in the body. Even if they're not growing and dividing, they're still actually actively manufacturing and secreted proteins.
Exactly. I mean, this changes the understanding of gut feeling, for example. That
Of what feeling?
Of gut feeling, you know, that they, yeah, I mean, that the probably our cells maybe can talk to microbes, etc. You know.
Oh, yes, of course. Yeah, there is active communication between the microbes in our intestinal epithelium, and they may play a role not only in normal physiology but in the generation of disease. I've become very interested in Parkinson's disease, and one theory is that certain gut microbes may initiate the accumulation of a protein called alpha-synuclein that aggregates in cells, and it may be conveyed from cell to cell up the vagus nerve into the brain. That's one theory about the origin of Parkinson's disease. And it relies on some kind of perhaps vesicular communication between a gut microbe and the intestinal epithelium.
Exactly. I mean, this opens another area in understanding the health and physiology.
Mm-hmm.
Yeah. So this work was appreciated by people in Stockholm in 2013 with James Rothman and Thomas Südhof.
Mm-hmm.
So what has changed after that?
Well, I've gotten older. The work goes on. We're still interested in vesicular traffic. Much of my work, even before then, shifted to studies on human cells growing in cell culture. And now we're working on another less well understood form of vesicular traffic that involves cells manufacturing vesicles that are exported outside of the cell, so-called extracellular vesicles or exosomes. And these are interesting because they contain RNA. Normal secretory vesicles don't contain RNA, but extracellular vesicles do. And there's some evidence that these RNA molecules contained in extracellular vesicles may be delivered to a target cell and change the metabolism of the target cell, or these vesicles may be exploited in disease for the spread of disease. There's some evidence for a role for exosomes in the spread of metastatic cancer. So it's become a very important area in physiology and disease. And so I'm quite excited about that new area that we're working on.
Yeah. So one important distinction between two processes is that in the first one, the molecules are directly secreted in the extracellular matrix. In the second one, they are secreted, or the RNAs are secreted in the exosomes, like in the membrane, right? And
so that means that in order to be taken up by a target cell, the extracellular vesicles have to be internalized, kind of like a virus particle, and there must be some membrane fusion event between the exosome membrane and the endosome in which it's housed, so that what's inside the exosome gets exported into the cytoplasm of the cell. And there's really very little known about that. It must happen, but very, very little known about it. So exactly. I like this area because it's important, it's active, and yet there isn't all that much basic biochemistry and cell biology that's being done.
Yeah, so in that case, a long distance cell, not only can change the I don't know, certain gene expression, etc., in the other cell, but it can also change the membrane composition.
Yes,
because, right?
In principle, it can.
Yeah.
But very few people have really studied it in that depth. But I think the field is going to grow and mature, and there'll be more people with a mechanistic appreciation who join the field and answer these questions.
Yes, and the interesting thing here is, like briefly, your recent papers that they show that in most of the exosomes you found this popular microRNA 223,
yeah.
Which seems to be delivered from one part of the body to the other, right?
Right there, but the evidence that miR-223 is actually being delivered functionally to another cell is pretty weak. Part of the problem, at least in the cell culture models that people use, certainly that we use, is that miR-223 we've shown is very highly and selectively enriched in exosomes. But chemically, it's not very abundant, very, very sparse. And it seems hard for me to imagine that enough of that rare microRNA could be delivered to a target cell to change gene expression, since microRNAs are thought to control gene expression by dampening the protein synthesis of a particular gene. And so it would have, these microRNAs would have to act stoichiometrically rather than catalytically. And I find it hard to believe that. Now, it may be that in more natural physiologic settings, there are some avenues of exosome biogenesis that enrich particular microRNAs to a much higher chemical concentration, but we have not seen that in the cell culture models that we use.
Yeah, so let's hope that more people will work on, or start working on this and start.
They will, because there's an enormous interest in this area in biotechnology. I sit on 6 different biotech companies.
Wow.
That are interested in exosomes for therapeutic applications or for diagnostic applications. And so there's a great demand for trained personnel. So I'm sure that a lot of labs will be studying this in the future. So I'm happy to be part of that.
Actually, well, when I was preparing this session, I checked that FDA so far hasn't approved anything for the.
Oh, no.
Yeah.
There are lots of problems between discovery and application. One of them is generating enough material. These exosomes are not terribly abundant when expressed by cell culture or in a, in an experimental animal. And they're very diverse. They're not just one type. So I think that's going to be a bottleneck for the creation of a drug delivery vehicle based on exosomes.
Yes. Let's move on to your another important work, which is on scientific publishing. And let's start with this basic understanding that we need to understand how scientific publishing works, which is peer review.
Yep.
So what is peer review according to you, and how does it work?
Well, peer review is the evaluation of one's work by scholars of standing in the field. And in the past, it used to be all anonymous, where you'd send a paper to a journal and the journal would select appropriate experts to consult. And those people would render an opinion anonymously. Increasingly, though, people are being asked to identify themselves as referees. And if they're comfortable doing so, that's fine. But the worry is, of course, that if they're identifying themselves, they may not be as, they may not be willing to express themselves as freely. Now, that doesn't mean that they have to be nasty, but that they should be willing to be critical, fair but critical. So I think the peer review system is essential, and I don't think there's going to be any viable alternative to that. On the other hand, we've also seen in the life sciences now an adoption of posting of manuscripts before they've been peer reviewed on an archive. One's called the bioRxiv. So the bioRxiv has now become very popular, but one must remember that the work that's posted on the bioRxiv has not yet been peer reviewed, and so it may have mistakes or inaccuracies that can be corrected. But even when it's subject to peer review, there may be mistakes that persist, because peer review usually only involves a small number of individuals and they can't catch everything. So the bioRxiv is good because it gives a larger number of people an opportunity to see a work that has not yet been published, and gives them, if they're willing, the opportunity to express some concerns to the author. And the author may not have considered these concerns. So I think it's a good thing. But still, before it can be adopted and generally accepted, it really should be peer reviewed. Now, the problem that I've seen over the course of my career is that the job of organizing peer review used to be achieved by scholars in scientific societies who organize themselves to publish the work in their field. But increasingly, and I think unfortunately, the field has become dominated by commercial interests who see the value in exploiting, frankly, the vanity of scientists who like to see their name up in lights. And so the most selective journals have a marketing strategy and a branding strategy that has captivated the attention of scholars all over the world who desperately try to get their papers published in these journals. Frankly, these journals are managed by people who are in the business of selling magazines. They're not fundamentally, or they're not singularly in the business of promoting scholarship. And this, I think, has been a bad trend. And so I've spoken out very strongly and publicly against this. And this was part of the reason why I was willing to start a new journal called Elife, that is where all the decisions are made by active scholars. But in fact, most journals are run by active scholars. It's unfortunately, the most popular ones, particularly Nature, Science, and in the life sciences Cell, where commercial interests have taken over. And I think this has distorted the meaning of scholarship. Commercial interests are promote papers that they see as potentially popular. So they may have a very short half-life. They may be important at the moment, but they don't necessarily reflect the long, deep scholarship that it sometimes takes for papers to achieve recognition.
So then let's just talk about Elife, and then I think more or less that sums it up.
Right, so Elife was started, I had served, part of my philosophy formed while I was the editor-in-chief of the journal, The Proceedings of the National Academy of Sciences, which a job I took in 2006. And I took that job because I had a long history of publishing in the PNAS. And I remember it being very informative to me when I was just in college. And I really enjoyed the format and the breadth of scholarship. And here again, it was controlled entirely by non-commercial interests for the benefit of the National Academy of Sciences. It wasn't never intended to make money for the National Academy. And so I started, but I became really irritated with the influence of a number that had been promulgated to be used by librarians to make decisions about which journal to subscribe to. It's called the journal impact factor. And it's basically a crude number that measures the popularity of a journal. But it doesn't measure the impact of an individual paper. It just measures the popularity of the journal. And it had changed over many years into a measure that young scientists would know to 3 significant figures and make decisions about where they wanted to publish based on this meaningless number. And some of the staff at the PNAS told me that I should be rejecting more papers because that would increase the impact factor, giving it the appearance of being more highly selective. And that is precisely what I had no intention of doing. I think that's the wrong attitude. And so I railed against this, and a lot of other journal editors felt the same way. And so I participated in a charter for something called the Declaration of Research Assessment, DORA, which calls on everyone involved to move away from the use of that crude number. But it persists. I still see it all the time. I see it advertised, and it's pernicious. And it's been exploited, frankly, by the commercial interests to peddle their magazines. So it's a very bad tendency around the world, and evaluation committees are influenced by this. They're influenced by where something gets published rather than what's in the paper. And I think this is just terrible. And I fight against it. And when I won the Nobel Prize, I announced that I wouldn't be publishing in these journals. I made a very public statement about this. And people thought I was hypocritical because I used to publish in those journals. And now, of course, I could afford to do whatever I wanted. But that was, of course, the only time that anyone would listen to me. So I still feel very strongly, but unfortunately, it's that ship has sailed, and I'm afraid it's so deeply culturally embedded now that there's no turning back. Anyway, so in 2011, we started a new journal with support from the Howard Hughes Medical Institute, from the Wellcome Trust, from the Max Planck Society, that we hoped would be more effective in competing with these journals. And we had some editorial policies that were unique, and it has been very successful, but it hasn't supplanted the commercial journals. And I think that there's powerful forces at play here that make it difficult, if not impossible, to reverse that. And I think it's terrible, but I can only do so much.
And many people actually have this sort of misconception that the Elife has an impact factor now.
Well, it does, but it's meaningless. When journals start, you're encouraged to apply to the organization that computes this number. And we, of course, had no intention of doing this. We did not apply, but we were informed that they were collecting the data anyway, and that they would compute our number. And even when we asked them to leave us alone and not do this, they said, it's public information, we'll do what we want. So yes, there is an impact factor. I don't know what it is. I don't care what it is. It's meaningless. but there it is. And some people will look at that and say, oh, I'm not going to publish there because its impact factor is low. Well, you know, there's only so much I can do.
Yeah. Okay, so let's hope that the trend will change.
Yeah, I don't know. I'm not sure what will. There are things at play that expose the commercial journals for the avaricious pirates that they are, but I don't think that will change anything.
Yeah, I was in a conversation with a scientist who ranted about the journals. And then when I asked him that if tomorrow you wake up and you get to know that you found your most successful discovery of your life, where will you publish it?
Yeah.
And he's like, yes, it will be one of Nature, Science, or Cell journals. So
yeah, that's because scientists are vain. They like to win the lottery.
yeah,
And that's what the commercial outfits appreciated. And they built huge financial empires on this basis. Elsevier is the largest publisher of science in the world, and they have a profit margin that exceeds essentially every other corporation in every other avenue of business, because they rely on the free services of scholars to review the papers. They charge for the publication of the paper, and then they turn around and charge libraries to subscribe to the journals. They're making money hand over fist without doing what a scientific society does. I'm not going to change my mind, and I work very hard to convince others, but it's been extremely difficult to do so.
Thank you so much. Yes,
luck.
take care. Have a nice weekend. Yeah.
Yeah.
Bye.
Bye.